My name is Yudi Xu. I'll introduce the paper submitted to iMeta. The title is Immune-Related Interaction-Perturbation Networks Unravel Biological Peculiarities and Clinical Significance of Glioblastoma. Glioblastoma is endowed with an infiltrative growth pattern and inherent difficulty in treatment, which could partly attributed to tumor heterogeneity. Thus, molecular classification is needed to decipher glioblastoma heterogeneity and facilitate individualized therapies. Previous researchers developing subtypes were based on bulk RNA-seq data, which ignored the fact that biological systems are dynamically altered. Conversely, biological networks containing the information of genes and interactions are relatively stable to time and conditions. Cancer cells in glioblastoma generated a pro-angiogenic and inflamed microenvironment, which recruited immune cells and molecules to infiltrate the tumor mass, leading to intricate immune networks in glioblastoma. An immune-related interaction-perturbation framework was introduced, which both considered vital interaction information in the biological network and the relationship between immunity and cancer. Firstly, we conducted subtype discovery from the immune-related interaction-perturbation matrix. Based on the interaction-perturbation matrix, 532 glioblastoma samples from the iMeta RNA-seq cohort were divided into 4 groups via consensus clustering. The survival analysis suggested a significant difference in prognosis among 4 subtypes, in which C4 displayed the most prolonged overall survival. Then, we conducted subtype validation in external and internal cohorts. The external cohort was the iMeta microarray cohort, and the internal cohort was the ZZU in-horse cohort. The 2 validation cohorts demonstrated similar transcriptome and clinical traits with discovery cohort. 4 subtypes also maintained comparable proportions across different cohorts. Then, we explored biological peculiarities underlying 4 subtypes. The result demonstrated that the C1 was defined as the immune-infiltrated glioblastoma, C2 was defined as the invasive glioblastoma, C3 was defined as proliferative glioblastoma, and C4 was defined as synaptogenesis glioblastoma. Then we explored the immune landscape of 4 subtypes. The result of immune landscape demonstrated that C1 tumors might benefit more from immunotherapy. Then we explored the immunotherapeutic potential of 4 subtypes. The result demonstrated that C1 may potentially respond to immunotherapy. Then we explored the genomic features of 4 subtypes. The result indicated that the 4 subtypes contained distinct genomic features. Especially, C4 was endowed with the highest IDH mutations corresponding to the favorable prognosis of C4. Finally, we explored the potential therapy agents in 4 subtypes. We detected subtype-specific interventions for 4 subtypes, which also concordant with their respective peculiarities. To sum up, an immune-related interaction-perturbation framework was introduced, which both considered vital interaction information in the biological network and the relationship between immunity and cancer. 4 glioblastoma subtypes endowed with distinct clinical outcomes and biological features were identified and validated based on the immune-related interaction-perturbation network. This taxonomy might be a promising platform to decipher the heterogeneity of glioblastoma and facilitate tailored management. That's all. Thank you. iMeta, a journal to change the world.